Chapter 5, Kinetoplasts, Trypanosoma and their kin. Part 14, Leishmania donovani. Leishmania donovani. You'll see this around the world. Different areas it can refer to as dum-dum fever or Kala Azar. When it comes to the morphology and the life cycle, what we're going to see is intracellular amastogotes. Intracellular amastogotes means we're no longer cutaneous. We've now moved on to what is known as visceral leishmaniasis. Visceral, meaning viscera, meaning internal organs are now going to be infected. Spleen, liver, mesenteric lymph nodes, bone marrow. What do all these have in common? They're highly vascularized. When you look at it, the life cycle. Sandfly ingest the amastogotes, which transform to slender promastogotes in the midgut, block the GI tract of the sandfly. So when the sandfly goes in for, the next meal, it regurgitates all the contents of its stomach as it's trying to spit in the saliva, in regurgitating into the wound, the promastogotes, which then go and are now in the bloodstream. Again, intracellular parasite gets engulfed, they get engulfed by the macrophages. It's where they want to be. They're protected from, other innate immune cells, and they're protected from the adaptive immune response. No B cell, no T cells. Asexual reproduction within the macrophages. Macrophage swells because of the internal pressure. All those parasites bursts, spreads, and that's how you see it going. Spreads to new host cells, and it just keeps going on and on. When the sandfly goes in for its next meal, or another sandfly comes in for a meal, it will take up the blood, which will have the amastogotes in it, or amastogotes in macrophages, and it just continues and continues. Clinical symptoms for donovani infection go from, asymptomatic. Somebody doesn't even know they're infected because it's at such a low level, and their immune system is able to keep it in check, maybe even able to clear it given time, to an extremely progressive. You start to see tissue damage, organ damage due to, the parasites infecting cells. Incubation from the moment of the bite could be anywhere from 10 days to a year before symptoms start to present. Starts off with symptoms that are very flu-like, okay? Fatigue, achy, feverish, and then it progresses. Instead of recovering, I think I got the flu, it just keeps going and going, and it gets worse and worse. And you start to see that these individuals will start wasting away, okay? All their nutrients seem to be going elsewhere other than their own tissues. Because of the extreme fever for such a prolongated time, they're burning calories that way as opposed to maintaining their own body. The anemia, red blood cells seem to be attacked. The macrophages are the host cell of preference. The red blood cells start to get damaged. So the person who's infected will start becoming anemic. Start to see that given time, Leishmania has a tropism, seems to like the liver and the spleen. And you'll start to get them enlarged as more and more macrophages, more and more innate cells start to collect there due to the presence of Leishmania donovani. Ultimately, it's going to progress to the point that they will die. Mortality rate is quite high if it progresses this long without some form of medical intervention. As you see from this young person here, their body has wasted because all their calories, all their energy resources are going to the prolonged high fever. And when I say prolonged, not just a few days, I'm talking weeks, months of a fever that literally burns through calories, lots of calories. And if you also notice the abdomen, it's got on the far left there, has that white tape, so you can see just how distended it is. That's not due to something like we have seen in previous parasites in this chapter, the megaesophagus, the megacolon. Now this is due to the enlarged liver and the enlarged spleen, which are swelling due to an overreaction of the inflammatory response, the accumulation of white blood cells, and the accumulation of the parasites in those organs, in those tissues within those organs. This is what's known as visceral leishmaniasis. Okay? Wherever there's highly vascularized tissues, liver, spleen, bone marrow, and whatnot, that seems to be where this parasite likes to collect. It gets taken up, gets its way into a host cell, because remember, it's an intracellular parasite. It's now hidden away from the immune response. It's in a cell that is filled with amino acids, filled with proteins, lipids, and all sorts of stuff that every growing cell wants, every growing parasite needs, and it starts going through binary or asexual reproduction, reproduce, host cell bursts, spread to new cells. Now remember that reproduction, asexual reproduction over and over and over, those rounds, that could take days, weeks for that to occur. So this isn't a very fast onset. This is a slow, chronic disease. You look here to an autopsy photo. Notice the spleen and the size of the liver. They're 3 to 4 times, well, that spleen actually looks about 5 to 10 times larger than it should be. And this is in a small body. So that's why the abdomen gets severely distended. When it comes to the immunology of this, the immune response is going to be different whether or not you're talking visceral or if it's still just in the cutaneous stages. Cutaneous stages, yes, there's going to be an immune response. The innate cells start to see the beginning of the adaptive response. But if it spreads and becomes a visceral, it's now intracellular. The innate cells are going to be the host, preferential host cells for this. And the adaptive response can only be, the B cells can only be of effect when it's already burst out of its host cell and it's spreading to its next host cell. So we're going to see also patient-to-patient variability is going to be an extreme issue here. Now, if they're subclinical, still in the cutaneous, okay, we're going to see a very strong Th1 T cell response, okay, which means you're going to get a pretty good, response here, going to get the innate adaptive response and a pretty good chance of clearing the infection. But if it becomes a prolonged infection and becomes chronic, and they become very symptomatic of, what we'll then see is a shift from Th1 to a Th2. And it's now more of a B cell, not so much the T cells that are going to be responding here. B cell response, great. You're going to have a high antibody titer, but the antibodies can only come out to play when the parasite is extracellular flowing in the blood. Great. You opsonized with antibodies all the parasites in the blood, but the majority of the parasites are still going to be hidden away in macrophages or endothelial cells lining, that reticular system. What we also see is we're starting to study and find that there are different phospholipids. What is it? Lipophosphoglycans So it's a phospholipid in the plasma membrane of donovani that has specific sets of sugar moieties on it, that these seem to inhibit gene expression of the macrophages. So, an evolution adaptation by Leishmania inhibits the intercellular responses of our cells. Diagnosis and treatment. Well, diagnosis, again, find the LD bodies, scraping of the vesicle, going in for, taking tissue samples, finding, doing, drawing blood, doing a blood smear, and finding, macrophages that are, infected with the A mastigotes. They're trying to develop, again, immunodiagnostic testing for presence of antibodies. Thing is, the antibodies are not always going to be species specific. They'll be specific for, here's proteins that are found on most species within the genus Leishmania, not Leishmania donovani specific proteins. And the thing is, we need to come up with better treatments, because you see here, this is a picture of a person post chemotherapy. They've had the drug treatments, and they have what in their area of the world is referred to as Kala-Azar. Okay, the visceral, luckily for them, they, whatever visceral damage was minimal, so they're still alive, but the mucocutaneous aspect of it, look at their face. The swelling should go down over time, but there will be scarring. There will be some underlying alteration of the tissue, so they're never going to look exactly like they did before the infection.